a long time ago, I did this film series at the library about food and its effect on the environment and stuff. And there were 9 films. And so no one in these, so the groups were interested in the films as events, but they didn't want to enter all the information. So they made me a co-organizer. Yeah.
We were talking about the other day where you're
Yeah, you know, I work over the hill, so it's a stretch for me too. No, these were in Santa Cruz mostly, and then some in Scotts Valley. I think we did one in Aptos on the weekend. Yeah. It was a while ago. Yeah.
search and replace, saving them.
Well, you know, like 15 people RSVP'd, and here we are, right? It's made up. Well, I tried to, but you can't win, really. People get mad at you if, like, not everyone shows up, and then people get mad at you if
We're like, man, we're really good.
If you're doing, yeah, so I don't think we've ever, like, had a place, not had a place in the race. I guess so. We've had, like, 40 people signed up, and we still, I think some people are on the tables, so they can yeah. So, but, you know, it's fun because you always have different people, and you get to know.
Yeah.
Oh, and they're very good, yeah. So which did you come with? Where are you from? Oh, I'm from the Bay Area. Bay Area.
We consider this today.
The East Bay. I guess not. Oh, you are. I mean, I went to
All right, if I can interrupt everyone on this lovely evening. It's about 7 p.m. That's normally when we get our Science on Tap started. Thank you all for coming out tonight. My name's Pauline. I'm the president of Women in Sciences and Engineering, and we host Science on Tap about once a month. We host Science on Tap about once a month at the Crate Place, and we're really thankful for them to let us do that here. Thank you all for coming out tonight. It's my pleasure to introduce Rita Mehta. She's in the Ecology and Evolutionary Biology Department here at UC Santa Cruz. Her lab focuses on functional morphology, and she's going to be telling you a little bit about moray eels, the aliens in our reef. So if you could give her your full attention, that would be awesome. Welcome her with a round of applause.
Thank you so much. Can you hear me if I don't use a microphone?
Really?
No. Okay. Let's see if I can.
Sounds like it's on. How's that sound?
Okay. Can you hear me better? Yes. Okay. Good. All right. Well, thank you so much, Women in Science and Engineering, for inviting me tonight. I'm very excited to tell you guys about some of the discoveries that we've made of studying moray eels off the coast of California. So, as Pauline said, I'm an associate professor in the Ecology and Evolutionary Biology Department, and my lab studies the form and function relationship in organisms. And I'm personally interested in organisms that live at the extreme. So hopefully that'll come out when you see a little bit more about moray eels. So I'm located actually not on the main campus, but on our really beautiful coastal science campus. The main campus is absolutely gorgeous as well, but we're so lucky here to have this other campus that's about 2.5 miles away. And we share this campus with the Institute of Marine Sciences, with Younger Lagoon. We are in a new building called the Coastal Biology Building, and the Seymour Center is also located there. So at the Coastal science campus, we also house a world-renowned scientific diving program directed by Steve Claybush. So one of the greatest joys of my job is actually getting to work with graduate students. So I have 5 graduate students in my lab of all varying years, so varying degrees in their, varying levels in their program. So Ben Higgins, who's here at this table right there, he's been helping me a lot with this moray eel study, and he's in his 6th year. Sarah Kinley studies phocids, and she is, seals and sea lions, and she is in her 5th year. Chris Law, who's also here, studies the evolution of mustelids, so raccoons, badgers, weasels, and he's very much interested in studying skull evolution of those organisms. Kelly Voss is a behavioral ecologist who studies octopus behavior. And Catherine Dale, who I know is also here, is interested in larval marine fish recruitment. So it's been a really great joy of mine working with all of these students that are interested in very diverse topics. Okay. Sorry, that's an awkward transition, but so one of the things that we do, and what's been very great over the last couple years, I'm wondering if there's a way I can make this not. Can you guys hear me okay? Okay. The entire lab at one point in time or another has come down to Catalina Island with me to help me study morays. And so what we do is we just drive down to Southern California, and we take a ship, which is the Miss Christie, through the Southern California Marine Institute. So as long as you're doing research on the island, this vessel will take you there from for free. It's a Coast Guard certified vessel, and every summer, at some point, you know, we take big action packers full of our dive gear, our personal belongings, and a bunch of food from Costco to help sustain us for a couple of weeks. So, how many of you guys have been to Catalina Island? Okay, great. So you know that it's one of the Channel Islands. It's actually not an island that's part of the National Marine Sanctuary. There's a pretty good population that lives on Catalina Island, and they're mostly in Avalon. There's a couple hundred people that live off of Two Harbors, which is on the west side. So most of you are probably familiar with the cute little bustling town of Avalon. As you can see, one of the big tourist attractions is the casino, which is now a movie theater, so it's no longer a casino. Avalon is filled with lots of different things to do. There's a beautiful botanical gardens, there's miniature golf, there's wonderful diving, and there's wonderful fishing. So we're actually not anywhere near Avalon. We're on the other side of the island, on the western part, called Two Harbors. So to get to, from Avalon to Two Harbors, it would be a 3-hour, very bumpy bus drive that most of us have done at some point or another and have vowed to only do it once. And then you could kayak from Avalon to Two Harbors, which we haven't done. Maybe we should try that one year. And that would be a 14-hour kayak, so we would be taking lots of breaks. And we probably couldn't carry all of our gear, but that's okay. So one of the greatest things about Catalina Island is the wildlife. So it's this beautiful scrub oak island, and it has around 61 endemics, many different endemic plants, and about 5 different mammal species. About 13 different species of birds are actually endemic on the island. There is the Catalina Island rattlesnake, which I just learned that someone at the LA County Museum is studying. And of course, it's known for the bison. that were introduced on the island in the 1920s for a big movie. And unfortunately, the movie tanked, and they basically just left the bison. And the bison, the bison are thriving there. So you tend to see bison when you're on the island at least once. So where we get to go at Two Harbors is a little cove over, and we actually work at the Institute for Environmental Studies, so there is a research station there. It's owned by the University of Southern California, and as you can see here, I'll be showing you some more up-close pictures. It's a really beautiful research station. When we had money way back when, like 6 years ago when my labs first started, it was so hard for us to convince them to let us do our studies there, just because we were not part of the Southern California Marine Institute. But we finally got our foot in the door, and after 6 years, now they just expect us to come, which is great. We've finally gotten into their good graces. So this is the dock that we would arrive at the Wrigley Institute. And as I said, it was hard for us to first get our foot in the door, but now we feel like it's kind of our second home in the summertime, so it's really good. And one of the reasons that it is so hard to try and get space there is because it's basically this consortium of 9 universities, mostly in Southern California, that pay dues every year. And the dues, I've learned, are pretty hefty. They're around $10,000 per institute. And what that buys the institute is they can have students come and do research on the island. It pays for the upkeep of the Miss Christie, so they can have free travel to and from the island. And we have access to the waterfront, which has these really great small boats, and there's a small boating area that we can use these in. And so all of these boats are maintained by USC as well as the Southern California Marine Institute with those shares. So right around the Wrigley Field Station is a marine protected area. And when we first started doing our research here, we didn't get permits to work anywhere in the marine protected area. So the places that I'll be showing you are mainly in these outer coves. But there's a reason why when you get on the dock and you look down, you immediately want to jump in the water. There's so many beautiful fish. And there are people that are monitoring the health of all these organisms down there. So, for example, one of the first things that you see, of course, is our California Garibaldi, and this is an adult, so our native fish. And you also see the blue-spotted juveniles that are really beautiful. We also see leopard sharks, and for a while there was a graduate student at Cal State Long Beach that was tracking the leopard sharks. That's another thing that's really great about the Wrigley Station, is that there have been a lot of tracking studies over the last 15 years. So we know a lot about the native animals in that area, or the native fishes. The blue striped goby, that's another fish that switches sexes. Ever since I have been familiar with the Wrigley Station, like for the last 12 years, someone or a bunch of researchers have been working on the blue striped goby. That's a model system for many different things. Hormone changes, so looking at their endocrinology, looking at their genetics, looking at aggressive behavior and territoriality. And then someone is also tracking the kelp bass. So as you can see, there's a really good, healthy population of fishes that you can easily see just by looking down from the dock. And it's a great place to take visitors, so people coming from the mainland. There's always a summer camp that will take kids snorkeling and kayaking. Many of these kids are just encountering the ocean for the first time, so it's a really wonderful opportunity to get them excited. When you walk up the dock, what you're going to see is another part of the waterfront, and there's a couple of tanks here and some holding pens, and this is ideal for doing invertebrate experiments or holding fish for short periods of time. We've tried to hold morays in there, but they escaped, and that'll be another story. But one day we're going to figure out how we can put morays in this place. So when you keep wandering up, what you see are research labs, a seminar room, a library. So, oops, sorry about that. All of this is very well outfitted with computers that you can use, printers. There's alumni cottages. And if you walk further up, there's dorms, apartments, and a cafeteria. And so I'm standing up on this ridge that's about a quarter of a mile away, and it's on the road to Two Harbors, which is the nearest town, and it's about a 2-mile walk to Two Harbors. So when you look out, these are some of our field sites that are out of the marine protected area. And you can see these coves all have a name, and we work within the coves and also between the coves. So these swaths here, I won't be talking about much, but Ben and a group of divers did some really detailed prey surveys right between these coves. And all of our trapping takes place along the walls of the coves. So when you descend down into the water in one of these coves, this is what you're going to find. So hopefully you guys see some lobsters down in these cracks. And it looks like there's one moray, but do you guys see the other moray that's right behind it? Yeah, definitely don't stick your hands in those cracks. So usually the visibility is pretty good. It's about 30 feet in the cove. Alright, so now you see the two morays looking right back at you. Alright. Yeah, you can, in this photograph, you notice they're really sharp intermaxillary teeth. So, we've been studying these aliens, and I need to give you some background to better understand why we call them aliens and why we were interested in studying moray eels in the first place. So one of the things that I mentioned earlier is, oh, okay, that's okay. Is that better? Yeah. Okay. So one of the reasons we study morays is because they're an example of an organism with a very extreme body shape. So they're convergent with snakes, right? Very long and skinny. The other thing that we noticed is that there's very little known about them. And I started out trying to better understand their morphology in the laboratory. So when we think about morays, it's better to take a step back and understand what exactly are they. Well, they're a bony fish. And bony fishes comprise one of the largest groups of vertebrates, or the largest groups of vertebrates. There's 28,000 species. And within the bony fishes, morays are one group within the anguilliforms. the anguilliforms are the true eels, and there's roughly 800 species of true eels. And you can see that they're really diverse. They live in the sand, they live in grass, some even live in algae, some bury themselves, some live in the rocky reefs, like you saw with the California Moray. They're about 100 million years old as a group. So we did a phylogenetic study in 2013, and we tried to get as much tissue as we could to understand the relationships of these guys. And one of the big findings that came out from this study was that they're a relatively old group. Their close relatives are bonefish and tarpon, which are really famous sport fish. And if you look at the shape of bonefish and tarpon and in any of the anguilliforms, you might be wondering, how the heck are those guys related, right? They look nothing like one another. Well, the big uniting feature that unites them is this leptocephalus larvae. So bonefish, tarpon, and all eels have this type of a larvae, that looks nothing like the adults. And essentially, this larvae gets to be bigger than what metamorphoses post-settlement. So the leptocephalus larvae are these big gelatinous leaf-like things that grow, and they are quite long-lived, up to about 3 months in some species, I think a little bit longer, some of the deep-sea species. And they just float until they figure out a place to settle. And so I don't think Pat would know if anyone really knows what is the allure, like what allures them to a particular settling spot.
Sometimes magnetism for some species of eels, but it's like very unclear.
Okay, so sometimes magnetism for some species, but it's otherwise unclear.
Yeah. Plug, I like it. Thank you.
Okay. So that's how we can put morays in a bigger context. They're a type of fish. They're relatively old, late Cretaceous. And why do I like them, right? Why do I like them so much? Well, I think they're really interesting, and for me, they've become a very interesting model because they do something different than what all other fishes do. So remember me telling you there's roughly 28,000 species of bony fishes. Well, all bony fishes, as far as we know, rely on suction for prey capture, for prey transport or swallowing, and respiration. So what is suction? Suction is essentially rapid movement of the skull to generate a pressure differential. And what results from this pressure differential is a flow of water moving into the mouth. For prey capture, within that flow of water resides the prey item. So this is an example of a juvenile tarpon suction feeding. And again, it's a very rapid behavior. So I'm using high-speed video to capture this behavior, around 500 frames per second. But I'm slowing it down so you could enjoy the feeding behavior. And it's slowed down to about 25 frames per second. Right. So you could see there the tarpon just rapidly expanded its mouth, right, to engulf the prey. So it was able to take in its prey without even touching it. All right, and one of my colleagues, when I was a postdoc, modeled the skull of fishes as an expanding cylinder. And we think that that's exactly right. The skull of fishes is a cylinder that rapidly expands, and that expansion is caused by over 50 different musculoskeletal elements in the skull. So fishes have the most highly kinetic or movable skulls of all vertebrates. However, when you look at a moray eel skull, it really doesn't look like your typical fish skull. most suction-feeding fishes don't have teeth. And that's one of the first things you notice about this specimen, which has been cleared and stained to expose the bone in the skull. So there's a little bit of blue here for cartilage, but all of this red means that this is all calcified or mineralized bone. So what you see here are the opercular series. And what's interesting about the opercular series is that they're really reduced. Now, I know you don't have a comparison, so you just have to trust me on this, but they have very reduced opercles. And one thing I can say that might help you understand what the opercles do is the opercles are those flaps on the fishes that are like little doors that open up when they're suction feeding, and they let all that water through to get that water out of their skull. The other bony element that we know from many studies helps to generate suction is a bone called the hyoid bone. Oops, sorry about this. So the hyoid bone is this little tiny bone right down here. And usually in fishes, this bone has a much thicker cross-sectional area, which also means that the muscle attaching to this bone is usually a lot thicker. And when I studied the muscle on the ventral side or the bottom of the mouth, what I saw is a very, very thin muscle sheet. So it makes sense that this fish is not generating suction. So what is it doing? Well, it's probably very obvious. It's probably very obvious with the teeth, right? Is that if you were to watch a moray try and capture prey, it's going to grab it by biting. But the perplexing thing is, although a moray might capture prey by biting, if it doesn't use suction, and most fishes also use suction to swallow their prey, how is it swallowing its prey, right? How is it able to get its prey from the anterior portion of its jaws back into its throat, right? And then into its esophagus. It kind of has a far way to go. So how many of you guys know, just out of curiosity, that fishes, all fishes, have a second set of jaws in their throat? So all fishes have a second set of jaws in their throat, and it's pretty amazing. So where did this second set of jaws come from? Well, what I did here with this tarpon is I removed the opercle so you could see all the gill filaments, right, the respiratory organs, and these gill filaments are connected onto the branchial arches. Well, of all fishes, the fifth branchial arch has been modified to become that second set of jaws. And what's interesting is when you skin a moray, because morays have skin, not scales, you see that the branchial arches and the gill filaments are behind the head. Whereas in this tarpon, the gill filaments are within the head area. When you do a radiograph of this moray eel, do you see these teeth that are right behind the head? Well, that's the second set of jaws. So one thing that I think would be helpful for me to describe is, what does the second set of jaws do? Well, for most fishes that are using suction to suck in their prey, and then to continue that suction force to push the prey into their throats, they'll meet up with that second set of jaws. That second set of jaws has teeth, and those teeth will usually pack or grind the prey and then push the prey into the esophagus. So it's another set of teeth that helps to break down prey and enables fishes, suction-feeding fishes that usually don't have teeth in the oral jaws, to have teeth somewhere in their throat region, right, to help them chew prey. All right, so what does a moray do with its pharyngeal jaws if it already has teeth in its oral jaws? Well, what you're going to see here is a slowed down video. This again was taken at 500 frames per second, and it's slowed down to 60 frames per second. And someone is holding a piece of squid really tight. They don't want the moray eel to grab this, because we want to see how that moray eel swallows its prey or transports its prey. So look just under the eye.
Wow.
All right. I'm going to play it again just in case you missed it. But keep paying attention right below. Oh gosh. I am so bad at this clicker. Oh my gosh. Okay. So keep paying attention right below the eye, right here. All right, so you probably guessed it, right? So the moray eel's second set of jaws is used to help it transport prey. And I'll show you in a bit. It's got sharp teeth on the pharyngeal jaws that basically ensnare the prey again and then move it into the esophagus. So it starts out behind the head, and then it can protract as far as the anterior portion of the eye. And this is just to show you that since all fishes have that second set of jaws, this is the diversity of the pharyngeal jaws that you see. What I'm showing you is the lower plate. So all pharyngeal jaws, because they're jaws, have two systems, right? Lower teeth and upper teeth. The lower teeth tend to have a broader platform, and so this picture gives you a better idea of the diversity on the teeth and the shape of the pharyngeal jaws. This is the moray pharyngeal jaws. Looks nothing like those, right? It's not going to be chewing or pulverizing anything, right? These actually look like little talons, right, that just grab onto the prey a second time. This is a scanning electron micrograph, so you can see how recurved the teeth were. I use this technique because in the literature, it suggests that morays envenomate their prey. So I used SEM to try and look for grooves in the oral jaws and in the pharyngeal jaws, and there were no grooves at all. So I don't think that they're envenomating their prey, or at least this species wasn't. So now, after I figured out what was going on or how moray eels use their pharyngeal jaws, I was able to capture some better video, or I had some more defined goals. So here, it's a little bit fast, but you could actually see the upper and the lower jaws. So I'm going to play it again. You guys see that? It's like a little tweezer, right? Coming up and pulling the prey back. And then this is my last video of the pharyngeal jaws, and this one I thought was a success because I was able to get a view, like an oblique view, so you could kind of see down in the throat, and you could see the anterior part of the upper and lower jaws come up. So, of course, that's a really neat way to swallow your prey, right? I think anyone that's interested in animals will think, wow, that's just a cool invention what the moray eel does, right? But I think stepping back and looking at the bigger picture in terms of organismal design, there's like 60,000 species of vertebrates, and there are so many different prey capture behaviors that have evolved, right? There's all sorts of envenomation strategies, there's luring strategies out there, there's even something called, you guys have all seen the anglerfish putting a lure out in front of its head. Well, there's even something called pedal luring, where a big frog in South America dangles its long toes over its head, and the toes look like little worms, so that a bird will come along to eat the worm, and then this Pac-Man frog will grab onto the small bird. I mean, there's so many just amazing different feeding strategies that have evolved. But when it comes to transport behavior, it's actually pretty boring. So there's suction, there's lingual transport, but this is a ballistic type of lingual transport. There's our lingual transport as well. There is inertial feeding, so like in a cyconiform bird, like a heron, something with a long beak, or an egret, they might probe into a hole, pick up a shrew, and grab it with the tip of their beak. But then how are they going to get that further into their mouth, right? You see them toss the animal, the prey item, up in the air and catch it further back. So that's an example of inertial feeding. There's gnathic transport, so gnathic as in G-N-A-T-H-I-C, jaw transport in snakes. And now this is a fifth mode of transport that's been discovered, pharyngeal transport. So in terms of prey transport behaviors, this is a new transport behavior in vertebrates. And so hopefully now you can make that connection with the title, right? Aliens in the Reef. So Carl Zimmer wrote an article for the New York Times, and when he was writing the article about the moray eels, he interviewed H.R. Geiger, the Swedish illustrator who came up with the alien. And Carl Zimmer asked him, how did you come up with the second set of jaws for the alien? Did you know about the moray eel pharyngeal jaws? And he said, no. In fact, my first alien that I drew for Ridley Scott wasn't scary enough. And so I had to go back to the drawing board, and I thought, what would be the scariest thing for me? Well, it would be a predator with two jaws. And so that's how the alien got the two jaws, and it's fitting for the moray eel. So we are definitely fascinated by these aliens, and we're fascinated because of the context of their morphology. They have some really extreme adaptations that would enable them to eat large prey. So a lot of people have asked me, well, do all moray eels have this adaptation? Do all moray eels have a second set of jaws? Well, since they're fishes, yes, they have a second set of jaws. There's roughly 200 recognized species of moray eels. Most of them are really, really colorful. They're found in the tropics. The hot spot is in Indonesia. And you can see just by looking at this, their oral jaw shape, or how long their jaws are, varies considerably. So they all have a pharyngeal jaw system. And we know that the oral jaws vary across species, but one of the things we started to look at next was, does the pharyngeal jaw system vary across the species? And what we learned is that body shape and diet does influence the oral jaws, and it does influence the pharyngeal jaws. So all morays have a pharyngeal jaw system capable of extreme movement. And what we discovered, so this is a graph showing number of vertebrae on the x-axis, and then the number of species that we looked at on the y-axis. So we just had a very small subsample. But the longer and skinnier you are, the farther back your pharyngeal jaws. And most commonly, species had their pharyngeal jaws around vertebrae 8. So what I'm going to do is I'm going to pass around the skeleton of a moray eel, of a California moray, and the pharyngeal jaws are attached to the skeleton. So this was nicely prepped by a postgraduate of mine, Samantha Gartner, who's sitting right there. And it shifted, so you can count. This one is probably between 7 and 8. It should be between the 7th and 8th vertebrae. So I'll start here so you can see it. So Rhinoneurina quasita, the ribbon moray, is the skinniest moray that we know of. It's a really beautiful moray. We know very little about it, although there is a paper that says that there are males and females of this species. We know very little about moray mating behavior, unfortunately. This is this is the skull that I'm going to be passing around the California Moray. And another thing that we realized is that there are morays that look to be piscivorous, right, eating fish because of these sharp teeth. But then there are morays with a skull design that is just so much more robust. The bone's thicker, this coronoid process to which muscle attaches is a lot longer. And there's even this prominence on the back of the skull that you don't see here that has extended the skull for more muscle attachment. And then look at these amazing teeth, right? These are molariform, or crushing teeth. So there's this type of dentition, and then there's some dentition that's kind of in between this and this extreme. But we know that this extreme is for eating hard prey. So we know that diet has shaped the oral jaws of moray considerably. And some of the evolutionary morphology work I've done, we've been able to show that durophagy, the ability to eat hard prey, has evolved over 13 times in moray eels. And we were able to get this result using a phylogeny. So what about the pharyngeal teeth? Well, it turns out that diet has also shaped the pharyngeal teeth of moray. So these are scanning electron micrographs of the pharyngeal teeth of different species. And one of the neat things that I want to point out is with the piscivores, you see these pharyngeal teeth are much more recurved, and there's kind of a lot of spacing to them. With the durophagus morays, those that eat hard prey, the teeth are not as recurved, but I think what's even more remarkable to me is that there's way more teeth on the pharyngeal jaws of a durophagus moray than there are on a piscivorous moray. And I think why that is because you're not ensnaring prey when you're durophagus. So what the pharyngeal jaws are doing is they're combing back fragments because the teeth on the oral jaws are actually crushing the prey. And then this is our ribbon moray that we don't know much about, but it's presumed to be a specialist on small crustaceans, and it's got those very small pointed teeth. So I wish we knew more about them because they are very interesting. So the morphological studies have been really fun to do, and they've given me a little bit of insight into the diversity of morays. I mean, there's a lot of species out there. They're found in mostly tropical waters, few in temperate areas. But I think what we wanted to do was push the question further and put it in a more ecological context. So essentially, are these morphological innovations that you see in morays, do they help them or do they facilitate consuming large prey? So these citations suggest that, but in these studies, no one actually looked at prey size that morays consume. So that's one of the things we wanted to better understand. We wanted to put our morphological questions and our morphological findings back into the ecology of the animal. And we wanted to understand how it related to the natural history of the California moray. So we wanted to know whether or not morays regularly consume large prey, what's the role of morays in the ecosystem around Catalina, and what's the density of morays around Catalina. So I'm not going to provide the answers to all of those questions, but I am going to talk about them. And one of the reasons that the California Moray is really interesting is because of its range. So this red line here is Point Conception. And what we see is that the California Moray, as far as we know, ranges down to the tip of Baja, maybe in the Gulf. We're still not quite sure. And there's a strong population around Santa Catalina Island. We don't know if there are many morays around the other Channel Islands. No one's looked.
They are in Santa Cruz, at least a few months ago.
Oh, really? Diving?
Yeah.
Fantastic. So this is when we embarked on our long-term study. In 2010 and 2011, we did this reconnaissance study in San Diego and Laguna to try and find morays. And we just used 6 traps, and we really did not find much at all. But then in 2011, we took these 6 traps down to Catalina Island, and we must have caught 12 in one night, and we didn't even use all 6 of our traps. So in 2012, we brought a team out, and now we have 6 years of this ongoing data. And as you can see, our team has changed from year to year, and the goals of each year have slightly changed as well. But the team leader for most of these years is Ben Higgins, who I said is my most senior graduate student right now. So Ben's been leading this big group for a few weeks down in Catalina Island each year. And it's a really great experience because we always bring undergraduates out, and these undergraduates get to put their scientific diving to good use. They also get to do some lab work as well as field work, and coupling lab and field work I think is really important and is really telling. As you can see, I was able to understand the morphology of the moray eel, but there's a limit to that when you're just in the lab, right? You also want to kind of bring it outside and understand and kind of ground truth everything. All right, so this is Ben and Kai, and 5 months ago, Rachel Higgins, Ben's wonderful wife, who's also been supporting us, had their second daughter. So, and as I pointed out, Ben is here now. So he'll be graduating with his doctorate in June of 2017. So he's got a spring defense, and he'll be talking about some of the data that he's been working up over the last couple of years. And I put this bold for Ben with the PhD because Ben has the silliest sense of humor. And everyone asks Ben, "You're graduating, right?" And Ben will look at me and he's like, "Yeah, I'm graduating with my PhD, right? Not just graduating the lab, but you're going to get a degree too, right, Ben? Hopefully."
Oh, you're a good Grace.
So this is where, again, it all starts. From the lab down at the docks in the morning. This is our 2017 crew, Kelly Voss and Samantha Gartner, half of our crew. And as you can see, here are our modified moray eel traps. They're custom made, they're anguilla traps, and we've worked with a really nice guy, Patrick, out in Long Island, who's helped make the holes bigger and everything so that it can accommodate the dimensions of a moray eel. Also, you might be able to see this water bottle in the trap. So the first year there was a little bit of trial and error trying to figure out how we were going to get the bait in there, but most importantly, we want to look at what the moray's eating. So we don't want to capture the moray eating our bait. We want to get morays that have eaten other things and have not eaten our bait but are attracted to the traps. So we have these water bottles that we poke holes in them, and we just stick a bunch of anchovies in there, 3 anchovies. And I think that smell is enough to lure the morays in. So pulling up traps is really exciting for us. It's like Christmas every morning. The traps get deployed in the afternoon. There's an average 16-hour soak time. And then usually we meet on the dock around 7 and start pulling up the traps. We start with our farthest site. And, you know, we can catch anywhere from 0 to, this was our most in any 1 year, 16 individuals in a single trap. So the traps can get really, really heavy. And once we get them on board, here you could see the water bottle with the poked holes, and this is, this pot has a ton of morays in them. But we open one of the doors, and then very carefully we try and shake like 3 or 4 individuals into a 5-gallon bucket that has some Finkwell, which is a anesthetic. So we just want to lightly anesthetize the moray. So this is an example of a really large moray in a 5-gallon bucket. And in this case, you can see what happens is they start to respire the anesthetic, and then after a while their respiration just gets slower and slower, and we can still see the heart beating, especially in a large animal like that. So we want to make sure that the moray is down, because we do a lot of morphological measurements, so we want to be confident that I can handle the head without getting bit. And sometimes, while under anesthesia, silly things happen, but what the moray doesn't know doesn't hurt it. And we usually work pretty fast. So there's a really good kind of conveyor belt on our boat. So what we do is we moor up in a cove where we've caught the morays, so that we always release the morays in that same cove. We measure body dimensions and mass. We measure gait dimensions, we palpate for diet, and we pit tag the individual. And so now, since we've been coming back so often, we are scanning a lot of these individuals that we capture to see if we have recaptures. And one of the things we've been doing is we've been keeping track of individual growth, and we've been also keeping track of whether or not individuals are constantly eating the same thing and how much their diet varies over the years. So this is an example of our data sheet, which is pretty fun. So you know, we've got all of these measurements. We've got a hideous 12 unit pit tag number. You've got the eel release site, which is usually the trap location. Gut contents, you always circle yes or no. And then occasionally, when the gut contents are really fun, like something pretty amazing, we end up saving it in a little vial, and then we write notes. And usually in our notes, if the prey item is whole, we will measure the prey item and put the measurements in the notes. Since we've been doing this for so long, what's been so exciting is now, even if a head of something pops up, we can measure the head and put it on our regression for head length and standard length, and we can pretty much get the standard length of the fish that the moray consumes. So one of the things that we realized is that morays have really large stomachs, and we think that this is to accommodate large prey or lots of maybe you know, small to medium-sized prey. So the gastric lavage technique that you use for most fishes by pumping water in the stomach doesn't work that well. And of course, we don't want to dissect every moray. That would be silly, because we want to understand what their role is in the ecosystem. So what we've been able to do is a technique called manual palpation, which is a technique that you do when you study snakes. Snakes have that convergent body plan with morays. And when a snake eats a large meal, you can actually see a bolus in the stomach. And very similar with a moray eel. So you can see that this animal ate something pretty big, and it's tapered, this item, so the head's this way, the tail is this way. And we usually don't have a lot of trouble trying to get the prey item. Sometimes the pharyngeal jaws do make it a little bit harder, and they snag the prey item, but if we have forceps and we're really patient, we're able to get the prey item out. And there's been times, we know, like a handful of times, when it just is too hard and we just let it have that meal. That's fine. So after we palpate the animal, and we get, and we get all of our measurements. This is one of the fun parts, is we release them once we know that they're awake from the anesthetic. And this was an event where we had a bunch of divers watching the animals so that they could try and understand where the morays go. So where they tend to hide out, or what the moray's next move was after we put it back in the area that we trapped it. So you see all the divers are having fun taking photos, and it's going to leave here in a little bit, which is why I let this video go on. So it's a very graceful animal. Alright, so the dietary items over the 6 years, that's been really fun. The most prominent dietary item are the calico bass, so the kelp bass. Octopus tends to be the second most, and then red rock shrimp and other fishes. During El Niños, when you have the warm water, we tend to see a lot of these tuna crabs come up. Oh gosh. Sorry. Okay. Sorry, I don't know what is the problem. Yeah, sorry about this. It just kind of died on me. Okay, so with the diversity of prey items, one of the things we wondered is, how is the moray able to catch such a variety of prey, from small lobsters to tuna crabs to the kelp bass and to octopus? And again, one of the great things about morays is they have a very diverse behavioral repertoire. So morays have been shown to shake their prey, knot around their prey to get some purchase, ram their prey into something, or rotate their bodies really quickly. And I had an undergraduate researcher with us one summer who was really interested in moray behavior, or just behavior in general. So one of the things that she proposed to do for her senior thesis when she got back to UCSC was to look at prey handling behavior in the California moray. So we were able to bring up 8 moray eels, and she was able to use 6 of these for her project. And she looked at how feeding dead fish and octopus of varying sizes affected the prey handling repertoire. So that was her senior thesis. And it was a really cool senior thesis. And she had some neat behaviors of the California moray that she was able to document and put with the publication. So this is an example of knotting. This is knotting in slow motion. And this is an example of shaking behavior. So this is shaking in slow motion. she also was able to observe rotational feeding as well as ramming behavior. But in the end, what she was able to find is that these prey items, when they increased in size, did affect total feeding time. And total feeding time was the addition of prey manipulation time as well as swallowing time. So the interesting thing that came with this study was that the type of behavior that they used did not vary with the prey. So they used shaking, ramming, rotational feeding equally across the prey items. But when the prey items got larger, they tended to use any one of those behaviors more. And they didn't use those behaviors with very small prey. The other thing that she noticed is that fish prey had longer manipulation times, whereas squid prey had longer swallowing times. So even though total feeding time did increase for both of these different prey items as the size increased, what we saw is that the component of different components, of total feeding time differed. So fish had longer manipulation times, squid had longer swallowing times. And what we attribute that to is the different material properties of the prey. So squid are really squishy and incompactable, whereas fish are really tough and they're hard to break apart. So another undergraduate was interested in doing a senior thesis, and he decided to look at the scaling of teeth and prey size in the California Moray. So Jacob Harrison had come down to Catalina with us 2 summers in a row, and we ended up keeping 21 individuals that were between 333 millimeters and 1,100 millimeters in length. And he decided to look at the ontogeny of tooth dimensions, so how these teeth, or whether these teeth change shape over time, which could give us insight into whether their diet changed as they got bigger. And what he discovered with the tooth patterns is that they pretty much grew isometrically, so all those different teeth in the jaws maintain the same shape, but the outer and inner maxillary teeth, which is what you see that's circled, those actually got larger as the morays got older, or got larger in size. And then when you look at prey size relative to moray size, which was one of the first questions we were really interested in understanding, we see that there's no relationship. In fact, when you look at the log of head length, or head length log transformed, and standard length of the prey item, you see fishes are in black circles, and invertebrates are in the gray triangles, and there is no relationship at all. So morays are basically opportunistic when it comes to prey size. One of the really great results, I thought, from Jacob's study was that he discovered that the oral jaws of the California moray teeth are retractable. So again, these 3 really long intermaxillary teeth, and there's always 3, tend to fold back. So they were always loose when we pushed on them. And Jacob discovered that, well, if you push on them just a little bit harder, they actually fold back to the roof of the mouth. So these really long retractable teeth are there to pierce the prey, but then when the pharyngeal jaws protract up to grab onto the prey, the teeth are able to fold back, and it loosens the grip so that the pharyngeal jaws can slide the prey back into the esophagus. Jacob is now in graduate school at Duke. So real quickly, because obviously I am really running over time here, and I don't want to bore you. One of the really fun things that we found out from this Catalina study is that there's a lot of morays where we've been trapping. There's 1,420 individuals. These are these have only been captured once, but we have many, many recaptures across the 5 coves. And this is standard length on the x-axis and frequency of occurrence on the y-axis. And one of the fun things that Ben did for part of his dissertation is he decided to age these morays so we could better understand how old they were in relation to their size. So he removed the otoliths from the skull, and these otoliths, as you can see by the scale bar, are just tiny. They're like little grains of rice. And what he found, there were two discoveries here. He found that there was a pretty strong relationship between the annuli, the rings that he counted, and the standard length of the moray. So I'm going to translate to you because this graph is head length. But essentially what we're looking at here is, this is our smallest moray eel. So I'm extrapolating the line a little bit. So this guy like barely fits around my neck as a necklace, right? This is probably about 3 to 5 years old, okay? But then we've got this guy here who's a little bit longer. So this is our average moray size. You could have the head and the tail touch around your neck like a charm, right? And so this is around 7 to 11 years old. So there's a lot of variance in that size range. This is our largest size range. So this is where you get your feather boa, right? This is like a nice big thick moray eel. And this can be 20 years or older. So, yeah. So that's one of our big discoveries from aging the morays. And this was really helpful because it really gave us some insight into the natural history of these animals. Also, what you'll notice here is that some of these points are in green and some are in red. Well, when Ben back calculated from the age of the morays, what he discovered is that the vast majority of them were settled during El Nino years. And so that also puts the density of Catalina Island into perspective, because what we're thinking is that these oceanic anomalies like the El Nino events are going to definitely, or are influencing the recruitment patterns of the California moray. So usually what we have is the subarctic current splits, and there's an Alaska current, and then down south you've got the California current. Well, when the trade winds relax, going east to west during an El Nino event, the Davidson current becomes more pronounced, and the Davidson current is in orange there. And what we see is that this seasonal fluctuation helps with recruitment patterns of larval fishes up to Catalina Island. So the moray eel is one of the fishes that benefits from this flow of water due to the El Nino event. So there's been a lot of benefits of conducting this long-term study. First is we've been providing a baseline for the population, and now we can look for shifts. And we've been engaging undergraduates in the field as well as in lab research. So that's one of the fun things about it, is that the undergraduates are able to, a lot of times, trap their morays and then bring them up to UCSC, where we have a marine lab, and we can hold them indefinitely and do lots of different experiments with them. And we can test these hypotheses in the field based on our morphological insights that led our interest in looking at the California moray in the field in the first place. And of course, the Wrigley Station is a really great place to work because not only are people very nice, but there's always researchers that the undergraduates can engage with. And there's this great poster there. I love it. I always hear undergraduates say, yeah, my parents aren't that supportive of me being a marine biologist or wanting to be a marine biologist. And whenever I see this poster, I think of that comment, because you can see how many Nobel Prizes have been won or have been awarded to researchers that have been at marine stations. The other thing that's been really great is helping the undergraduates figure out what they like to do. This is the last time I will have to do this. So I think that getting any type of research experience, whether or not in the lab or the field or both, can give you insight into career decisions later on. And I know that as an undergrad, that really helped me. So out of the 17 undergrads I brought down to Catalina Island, I've had one who was great in the field. And he wrote me when he got back and he said, that was really fun, but I felt like everyone else was way more excited about every single thing that we did than I was, so I'm actually not going to be pursuing this anymore. And that was great for him to understand that that was not what he wanted to do. But most of the other students have gone to graduate school, they've worked at NOAA or CDFW, or have helped with outreach programs afterwards. There's one now that's applying to go to medical school because he worked on this project because he just loves diving. So, and it gave him that experience. So lastly, and again, one of the benefits of doing these long-term studies is we finally got MPA permitting in 2015. And one of the things we noticed that was different from the MPA compared to all the other codes is these morays were, these are live animals, but they had these weird strictures down their backs. They couldn't straighten their bodies, but they could still swim. They were awkward. They were found in our traps. And in 2017, so just this past summer, we found 78 individuals like this, 27% of our catch in the MPA. So we know that the MPA has a much higher density, and we think that the morays there must be catching some sort of a bacterial or infection or something from the high density that there is in the MPA that may not be able to support them. So this is kind of this interesting self-regulation of moray eels, especially when we have more frequent El Nino events. All right, so thank you so much for listening to me ramble on. I'm sorry about the microphone. I kind of never got used to talking into it. But I appreciate your attention, and if you have any questions, I'm happy to try and entertain them.
Where do you get your funding? One question. Secondly, is this all reaching in sometime?
Yeah.
And maybe thirdly, are there other people like you on the other coasts or other companies?
Yeah, that's a really good question. So the funding issue, I've been able to get funding, especially early on. The University of California is really great with respect to trying to help their non-tenured professors get tenure and help them establish new research programs. So with some of the seed money that they gave me early on that I had to compete for through the Lucille Packard Endowment or from the Hellman's Foundation, I was able to get money. And, the work that I do it's not that expensive. So I'm really lucky, but it does take support. So with that money, I've been able to stretch about 3 field seasons. And then I had 2 NSF grants, and I was able to use some of that money, even though it wasn't proposed for this study, as seed money to get this going as well. But right now, I don't have any support for this project. The other question you asked is there an end? Yeah, I've been asked that a lot, actually. Is there an end? I think for us, there's so much of the baseline of the moray eel natural history that just still hasn't been established, but is now. We're publishing papers. So I think with the fluctuations in the weather patterns and climate change, now definitely would not be the time to stop recording data in this population. I can't exactly tell you how much longer, but I think the other way I see it is it's a really great tool for attracting and mentoring undergraduates in the lab in the field. And no, as far as I know, there are no researchers in other areas of the world that are doing this, although, hold on, I'm wrong. There is a woman in Sweden who's done some work around Greece. So Murena Helena is a moray that's been pretty well studied. But people have contacted me. You know, they've asked me to come out to Australia, or they've asked me to collaborate with them. And I think, you know, the hard part is time. There's just not a lot of time because I've got my own system that I'd like to work on.
What is the range of one moray? Like how far does it swim?
Yeah, that's such a great question. So our morays do not leave their coves. So we don't know how often they're swimming out of the coves, but we know that they're coming back to those same coves. And we have done some experiments where we've taken morays from Cherry Cove and moved them into Little Fisherman's Cove, and the next year we find those morays back in Cherry Cove. So they like being in their cove. But they probably like moving around. They probably end up moving around quite a bit for prey. We just don't know how much. Yeah, that's a good question.
Well, if we can give Rita another round of applause, thank you all for your questions. We hope to see you at one of our future Science on Taps. Thank you, everyone.
Thank you.
That was interesting. Where's the chef? I have to go to the bathroom one more time. All right, I'll wait for you. I'll see if you can get her tonight. I'll see
I have no idea. I can't find the stop button. Oh.